Hello friends, welcome to my course on microbial biotechnology. We're in module number 8, environmental biotechnology, where we are today going to discuss about microbial bioremediation. This lecture is broadly divided into two sections. In section 1, we will be having a small introduction to bioremediation. Then we will discuss about the major group of pollutants and try to find out what are the sources of environmental pollutants, and then the impact of environmental pollutants, why bioremediation is necessary, and the advantages of microbial bioremediation, and also discuss the potential risk of microbial bioremediation, and we will also discuss about the types of bioremediation, which are broadly of two types, in situ and ex situ. Microbial bioremediation is a process that utilizes microorganisms such as bacteria, fungi and algae to clean up and remove pollutants from the environment. And we also sometimes use plants, but that is dealt under a different subject called phytoremediation. Microbial bioremediation is a sustainable and cost effective approach to remediate various contaminants, including organic compounds, heavy metals and other hazardous substances from soil, water and air. So, the major group of pollutants can be discussed under the following heads. Number one is the BTEX, which basically stands for benzene, toluene, ethyl benzene and xylene. Mostly these are obtained or these are the pollutants which we get from the paint industry or chemical manufacturing and oil production. Then we have the pesticides, mostly which comes from the agricultural landfill sites and pesticide manufacturing units. And some of the examples are carbofuran, atrazine, diazinon, glycophosphate, etc. Then we have the polyaromatic hydrocarbons, which are basically obtained from the biodiesel and oil production process, then coke plants and gas work sites. Some of the examples include anthracene, naphthalene, fluorine, etc. Then there are the heavy metals, which are mostly obtained due to the agricultural activities, landfill sites and the mining activities. Some of the examples are lead, cobalt, cadmium and so on. Then we have the chlorinated solvents. These are mostly the outcome of chemical industries and dry cleaning process. So, we have some of the examples like trichloroethylene and perchloroethylene. Then we have the polychlorinated biphenyls, which are mostly sourced from the power stations and electrical equipment manufacturing industry. Some of the example include 4-chloro-biphenyl and 4,4-chloro-biphenyl. Then finally, one of the most dangerous ones, the radioactive wastes, which are the contribution of the nuclear industries, nuclear installations and nuclear weapons research, etc. and including nuclear power production plants. So, you have different isotopes of uranium and other metals which are very harmful to all of us. Now, what are the sources of environmental pollutants, or what are the sources of these environmental contamination? Mostly, as we have seen in the earlier slide, they comes from the industries like the plastic industry, even the wood, textile industry. The textile uses a lot of dyes, and then the preservatives, microelectronics, refineries, etc. Then we also have found out that agriculture is one of the point sources of environmental pollutants, mostly the agrochemicals. The excessive use of fertilizers, pesticides and weedicides are actually some of the reasons why our environment is getting polluted. Then the waste disposal in the cities as well as in the rural areas, the sewage, sludge, leach out from the landfills and fly ash disposal, etc. all contribute towards the environmental pollution. And one of the major contributor for environmental pollution are the mining industry. So, here smelting, river dredging, mine spoils and tailing and also metal industry, etc. contribute towards the environmental pollution. Now, what is the impact of the environmental pollutants and why we require bioremediation? So, here we now know the various sources of environmental pollutions as discussed in the earlier slide, and they actually impact the biological world and the environment, and they may actually pollute all soil, water and air. Now, they pose ecological and human health risk. They affect the animals, plants, microorganisms, and finally, these also directly impact human life. One of the problem with environmental pollutants is that we may have bioaccumulation. So, you can see that small organisms or small fishes are getting affected first, which are consumed by the bigger ones, and again by more bigger ones. And this is just a symbolic picture, but in the food network, smaller organisms are always consumed by bigger organisms. Therefore, when a lot of smaller organisms having small contaminations are consumed by a comparatively bigger one, the bioaccumulation takes place, and then finally it get biomagnified. So, you can see that from one organism to another organism there is a flow of these pollutants because of the food network, and you can see here the contamination level which is being shown by the size and the color. Now, if we do remediation, I mean treat these pollutants in the source or in the environment, we can actually stop these from entering the food network or ecological niche, and thereby we can actually save the environment. Bioremediation prevents these bioaccumulation and biomagnification of pollutants, thereby reducing ecological and human health risks while protecting ecosystems for long term harm. So, that is why this remediation is necessary. Now, what are the advantages of using microbes for remediation or microbial bioremediation? Number one is the safety. It reduces risks to human health and the environment. Then this is a environmentally friendly process. It reduces pollution without harming ecosystems. It is cost effective, lowers remediation cost significantly. And it is also a natural process which is enhanced. So, boost natural degradation process, then there is a minimal disruption, thereby preserving existing ecosystems during the remediation process, and it is having long term effectiveness and sustains remediation over extended periods, and it is a versatile process applicable to various contaminants and environments. Now, what are the potential risks of microbial bioremediation in spite of all the advantages? So, we have to consider these potential risks such as the introduction of non-native microorganisms and the potential release of harmful byproducts during the degradation process. Careful site assessment, planning and monitoring are essential to ensure effective and safe implementation of microbial bioremediation strategies. Now, what are the different types of bioremediation we can use for our purpose? The first one is the in situ bioremediation, where treating contaminated material is done at the site itself. So, this may be natural attenuation process, or it may be an engineered process where we go for bioventing, biosparging, bioslurping, phytoremediation and biostimulation, or it may be ex situ bioremediation where removal of contaminated material for treatment is done in a site away from the original location. So, this may be done by biopiles, windrow composting, bioreactors and land farming. Let us now discuss about the in situ bioremediation, which basically is a process of treating contaminated soil, groundwater or sediments directly at the site of pollution without removing the contaminated material. It utilizes natural or introduced microorganisms to degrade or detoxify pollutants in place or in the location itself. So, number one is the natural attenuation, which is the process by which naturally occurring microbes degrade or neutralize environmental contaminants without human intervention. This passive approach relies on biological, chemical and physical processes to reduce pollution levels over time. So, it includes the biodegradation or simply sorption, or we may also dilute the pollution, which is of course not a very scientific process. Then by the process of evaporation and also chemical reactions. However, regular monitoring ensures the conditions that support effective remediation is very essential. So, let us now discuss about the bioventing, which is basically a method of engineered in situ bioremediation. Bioventing enhances microbial degradation of pollutants, mostly petroleum hydrocarbons, by injecting oxygen into the unsaturated vadose zone, stimulating indigenous microbes to grow and degrade the pollutants in the site or location. Nutrients and moisture may also be added to optimize conditions for biodegradation, and there can be two types of bioventing. One is the active bioventing, where bioventing is generally performed using blowers. As you can see, this is blowing air through this pipe which is dig deep into the soil. So, which uses the blowing of air with the help of blowers. We call it as active bioventing. So, here you see this is the ground surface and this is the water table and this is the contaminated soil. So, we will inject or blow air through this duct with the help of a blower. And then we also have a monitoring system in place, which will keep on observing whether the biodegradation is progressing satisfactorily or not. This blowing of course is very slow. So, we do low rate air injection in the case of active bioventing. The whole idea is to facilitate the growth of the naturally occurring bioorganisms, microorganisms over there. In passive bioventing, we do not use a blower. So, here air intake happens passively through the passage we have built, and there is also a exhaust passage, as you can see over here, and then there is a vapor monitoring point to keep continuous observation of the progress. So, here in the passive bioventing, the exchange of gas happens through vent wells, and which is basically due to the effect of atmospheric pressure or barometric effect, or sometimes tidal fluctuations, and we do not use blowers here, and because of the absence of blowers, we call it as the passive bioventing. So, bioventing is commonly used for light petroleum spills. Its effectiveness depends on air injection rates, with lower rates often supporting better microbial activity. So, if we blow with very high speed, that actually will not help us in getting more efficient bioremediation. So, bioventing helps degrade volatile organic compounds. High airflow can sometimes lead to the release of these volatile organic compounds into the atmosphere, requiring additional treatment like biotrickling filters, which we have discussed earlier. Then there is the biospurging, which is similar to bioventing, involves injecting air into the subsurface to stimulate microbial activity for pollutant removal. However, unlike bioventing, air is injected into the saturated zone, causing volatile organic compounds to move upward into the unsaturated zone for biodegradation. So, the effectiveness of biospurging depends on soil permeability and pollutant biodegradability. Biospurging is used primarily for treating petroleum contaminated aquifers and effectively shifts conditions from anaerobic to aerobic, promoting biodegradation of pollutants like benzene, toluene, ethyl benzene and xylene. Briefly we call them as BTEX. Then there is another kind of bioremediation, we call it as bioslurping. This combines vacuum enhanced pumping, soil vapor extraction and bioventing to remediate soil and groundwater by providing oxygen and stimulating contaminant biodegradation. So, here you can see this vacuum pump, and then there is the upward movement of these LNPS, which are actually entrapped over here, and this is the ground surface, and this is the water table. So, these basically used to recover the LN APLS or the light non-aqueous phase liquids and remediate volatile organic compounds. The system draws liquids upward using a slurp, separating the LNAPLs from the water and the air. So, let us now see another process which we call as the phytoremediation. This is not exclusively microbial in nature. Phytoremediation uses plants and their associated microbial communities to remove, degrade or contain pollutants. Microbes in the rhizosphere around the plant roots in the soil play a crucial role in this process. So, here we have these rhizofiltration happening. The plant roots remove contaminants from water through absorption or precipitation. Then we have these phytostabilization. Plants immobilize contaminants in the root zone, preventing spread or bioavailability. Then we have the phytostimulation, where roots exudates stimulate microorganisms to degrade organic pollutants in the rhizosphere. Then we have phytoextraction, where plants, especially hyperaccumulators, absorb and store contaminants, enabling removal through harvesting. Then we have phytodegradation, where plant enzymes and symbiotic microbes break down organic contaminants. Then we have phytovolatilization, where plants absorb contaminants through roots and release them as volatile compounds into the air. Briefly, phytoremediation is naturally occurring, but when optimized for pollutant removal, it becomes an engineered bioremediation process. So, let us now study about the engineered in situ bioremediation process, which we call as the biostimulation. This involves enhancing the growth and activity of indigenous microorganisms already present at the contaminated site by providing them with nutrients, oxygen or other growth promoting conditions, and these accelerate natural degradation processes. So, this is the contaminated environment. So, here we will add nutrients. So, this will help the microbes to grow or multiply, and therefore also carry out the bioremediation, and then it thereby degrades the pollutants in the contaminated sample or site, and then we get a healthy environment. So, these are some representatives. These are the contaminants. Then these are some of the newly developed strains you can see over here, and these are the indigenous microorganisms. So, we may use both of them, and then these are the nutrients which we have added to enhance this process. So, this is in simple terms the process of biostimulation. Now, another approach for bioremediation is the ex situ bioremediation, which basically refers to the process of removing contaminated soil, water or sediment from its original location and treating it in a controlled environment to degrade or neutralize pollutants. So, one of the method is biopiling. Biopile remediation involves piling excavated contaminated soil and enhancing biodegradation through aeration, nutrient amendment and sometimes heating. This ex situ technique uses a treatment bed with system for aeration, irrigation, nutrient supply and leachate collection. So, here in this figure we can see a scheme of biopiling. So, this is the waste and we have a impermeable cover which actually is covering this waste, and then both from the upper side and from the bottom side. And then there is this gas water evaporator which will actually try to suck in the gas or the water, and that mixture will be separated. Water will be sent to a water treatment plant and the gas will go to a gas treatment or gas capture unit, and these water after discharge is maybe available for different use. And the water which is the waste and it is not could be purified. A part of it will be recirculated into the waste, and we also add microbes and nutrients into this waste from time to time or in the beginning itself. So, this is in simple terms a biopile, and this waste was actually excavated from or transported from another location and brought into this unit. So, here basically we may use indigenous microbes or naturally occurring microbes and sometimes engineered microbes. So, these biopiles are cost effective and flexible. This enables treatment in extreme environments like cold regions. Biopiles involve the following steps. Number one is the soil excavation. Second is the optimization of conditions like moisture, temperature and aeration, and also the control of these various conditions like air and moisture. Then continuous monitoring and harvesting of the treated soil. Once the soil is bioremediated, we will harvest that soil and we will use it for different economic purpose. Another method is the windrow composting. So, we can see here the rows here, these are basically all rows of deposited waste which has been transported or excavated from one site and brought over here, and we actually arrange them in parallel heaps, large heaps. So, windrows enhances degradation of hydrocarbons by periodically turning piled contaminated soil. This increased aeration distributes pollutants, nutrients and stimulates microbial activity, speeding up bioremediation through assimilation, biotransformation and mineralization. However, periodic turning may not be ideal for soils contaminated with volatile toxins and can lead to methane emissions due to anaerobic zones forming from reduced aeration. So, some of the steps that are involved in windrows composting, we excavate and screen the soils for bioremediation, and then we form windrows. Then we do periodic turning of the windrows, then windrow will be monitored and the compost will be analyzed. Then windrow will be disassembled and disposed of, and basically the soil is now ready to be utilized for some economic activity. Another approach is using bioreactors. These bioreactor based microbial remediation involves using a control vessel or bioreactor to enhance the degradation of pollutants by microorganisms. Bioreactors offer precise control over environmental conditions such as temperature, pH, aeration and nutrient levels, optimizing microbial activity and accelerating the bioremediation process. So, here we have this bioreactor unit with an agitation system. Then we have a channel for aeration, sending in air or oxygen, and we also have a feed inlet pump. Through these we will be sending in the waste to be bioremediated in this reactor tank. Then we also have sensor probes for monitoring of the bioremediation from time to time. And once the treatment is over, the effluent is actually obtained through a passage in the bottom of this reactor tank. Bioreactor is a versatile method. It can treat various pollutants in soil, water or air and is effective for both organic and inorganic contaminants. Bioreactors can handle toxic compounds and volatile organic compounds, making them suitable for treating complex waste. However, they can be cost intensive and require careful management of operational parameters to maintain efficiency. So, let us now look into the different types of bioreactors that are used in bioremediation. Number one is the slurry phase bioreactor. Here the polluted soil is treated and the sediment is obtained in slurry form. This is effective for petrochemicals and chlorinated solvents. Then we have the partitioning bioreactor, which uses two-phase systems for hazardous wastes, reducing toxicity to the microorganisms. Then we have the stirred tank bioreactors or STR, mechanically agitated, offering efficient gas transfer and mixing, ideal for submerged cultures. Then we have the biofilters, which use media beds for degrading pollutants, common in wastewater and air pollution control. Then we have the pack-baked bioreactors, provide fixed film bioreactors for high biomass treatment, often used in dilute aqueous solutions. Then we have the airlift bioreactors, which treat gaseous pollutants with air circulation, but limited by gas liquid mass transfer. Then we have the membrane bioreactors, which combine biological treatment with membrane filtration for high quality effluent, but can be actually very costly. Another method is the land farming. Land farming is a simple and cost effective bioremediation technique used for treating hydrocarbon polluted soils. Land farming involves excavating and spreading polluted soil from contaminate sites on a surface where native microorganisms degrade the contaminants through aerobic degradation. So, this is one schematic diagram of land farming. So, here we have these contaminated soil, and then there we do tiling for soil aeration, and then we have porous cup meters, and then we have a groundwater monitoring well, because the leachates may actually enter the groundwater sometimes. The leachate is collected and it is treated. Of course, that can be also optional, but it is always good to treat the leachate, which may be having a lot of harmful pollutants. The land farming include tillage for aeration, nutrient addition and irrigation, which stimulate microbial activity. It is effective for removing hydrocarbons via biodegradation and volatilization, and it can be applied in various climates with minimal environmental impact. However, land farming requires large space, is less effective for inorganic pollutants, and faces challenges with toxic volatiles and extreme climates. Let us now move to section 2, where we will be discussing about the various microorganisms used in bioremediation, like bacteria, fungi and microalgae, and what are the different factors which affect bioremediation. So, the microorganisms which are used in bioremediation include bacteria, algae and fungi. So, let us discuss about the bacteria first, which are crucial for microbial remediation, effectively degrading various pollutants. They assist in wastewater treatment by breaking down organic matter and heavy metals. They also degrade hydrocarbons in oil spills, synthetic dyes, and are also effective in breaking down pesticides and can detoxify heavy metals. Degradation of hydrocarbons by bacteria. Hydrocarbons, crude oil and petroleum, are broken down through aerobic or anaerobic respiration. Aerobic bacteria uses oxygenases, example monooxygenase or dioxygenase, to oxidize hydrocarbons, while anaerobic bacteria utilize alternate electron acceptors like sulfate. Some of the examples include Aphanocapsa species or Plectonema species for crude oil degradation. Then we have Acintobacter and Pseudomonas, and also Spingomonas for petroleum hydrocarbon degradation. Then we have certain bacteria which can degrade dyes, particularly azo dyes, through reductive cleavage of the azo bonds under anaerobic conditions via the enzyme called azoreductase. Aerobic degradation of the dyes involves oxidizing aromatic rings using monooxygenases and dioxygenases. Some of the examples include Aeromonas hydrophila, Bacillus subtilis, azodye degradation in aerobic conditions. Then the Pseudomonas and Proteus mirabilis, where anaerobic degradation of azodyes takes place. The bacteria are also used for degradation of pharmaceuticals and personal care products. These are broken down by microbes through complex enzymatic pathways. native bacteria metabolize or transform pharmaceutical pollutants into less toxic or non-toxic compounds, often aided by biosurfactants. Some of the examples include Pseudomonas and Bacillus. Bacteria can also degrade pesticides, particularly endosulfan malathion are degraded through hydrolysis, oxidation and reduction reactions catalyzed by enzymes like hydrolases, esterases and dehalogenases. Bacteria can use pesticides as carbon or energy sources. So, some of the bacteria that can degrade pesticides include Bacillus, Pseudomonas, Arthrobacter. And some of the heavy metals are detoxified by bacteria, particularly lead, cadmium, mercury via biosorption, bioaccumulation and reduction. Biosorption involves binding metal ions to cell wall structures. Reduction process convert the toxic metals chromium 4 to chromium 3 into less toxic forms. So, some of the examples include the D-sulfovibrio species, Geobacter species, where the heavy metal reduction takes place, and then we have the Escherichia species, Pseudomonas species, where methylation of mercury takes place. Let us now discuss about the role of fungi in the bioremediation. They are crucial in pollutant removal, especially heavy metals. By transforming them into less toxic forms, they are easy to cultivate, produce significant biomass and degrade contaminants like dyes, pharmaceuticals and hydrocarbons through extracellular enzymes and protective hyphae. Certain fungi in wastewater treatment include Pleurotus pulmonarius, then Aspergillus parasitica and Stachybotrys species. Then we have the lignolytic fungi with enzymes like lacase and peroxidases, which degrade complex synthetic dyes, and also yeast like Saccharomyces cerevisiae and Candida krusei absorb and degrade heavy metals or detoxify the heavy metals. Then we have the microalgae which are used in phycoremediation, which is basically bioremediation that uses microalgae for wastewater treatment. Species like Chlorella, Tetraselmis, Anabena and Spirulina are effective due to their ability to use inorganic and organic nutrients, and they are having short life cycles and can reuse the biomass. So, these are some of the species as shown here, Tetraselmis and Anabena, which are used in the bioremediation process. These microalgae can thrive in diverse conditions, remove heavy metals efficiently and release oxygen to support bacterial pollutant degradation. So, what are the types of microalgal systems that are used for bioremediation? We have the open systems where algae are grown in natural or artificial ponds and lagoons, often combined with bacterial cultures for effective wastewater treatment. Here we can see the marine Chlorella species cultivated in open pond over here. Then we have closed systems where microalgae are cultivated in control environments like photobioreactors, offering advantages such as reduced water evaporation and higher biomass yields. So, this is a photobioreactor system. Now, let us discuss about the various factors that affect bioremediation. It actually depends on the type of contaminant. Organic contaminants are more easily biodegraded than inorganic contaminants. Then also the concentration of the contaminant, high concentration of contaminants can overwhelm the microorganisms or create toxic conditions, and the environmental conditions like temperature affect the bioremediation process. Most microorganisms prefer temperature in the range of 20 to 40 degrees centigrade, and they prefer a pH of around 6 to 8, and oxygen basically aerobes are the primary microorganisms involved in bioremediation. Anaerobes are less common and less efficient in this process. And the presence of other contaminants also affect the process of bioremediation. Antagonistic interactions inhibit the activity of microorganisms degrading other contaminants. Then there are of course the synergistic interactions as well, which enhance the activity of microorganisms degrading those contaminants. Let us now go to section 3, where we will discuss about the recent trends in bioremediation, and then also we will discuss about the risk assessment of genetically engineered microbes. So, genetic engineering is becoming very, very important for bioremediation. So, let us first look into the bioremediation process when we use indigenous bacteria. So, here we have pollutants which are of high concern, but these indigenous bacteria may actually be not able to survive, or even if they survive, the survival may be low in case of high concern pollutants, and then they will not be able to remove these pollutants. So, efficacy is minimal and the degradation rate will be low or totally absent. Now, compared to these, if we use engineered bacteria which are basically produced by the process of genetic engineering, we may get high bacterial survival, maximal removal efficacy and high degradation rate. Now, what are these 4 steps of genetic engineering or engineered bacteria deployment? Number 1 is a screening and cloning of highly effective degrading genes in particular microbes which will be used for this process, increasing the expression of enzymes with degrading functions in microbes, then protoplast fusion by blending the advantages of 2 different microbes, then expression degradation genes for different pollutants, expression of degradation genes for different pollutants to develop super engineered bacteria. We can also go for genome engineering for bioremediation, particularly tools like CRISPR-Cas and TALEN enables precise insertion, deletion or substitution of DNA sequences in organisms genome, offering opportunities to enhance bioremediation. CRISPR-Cas9 in particular allows multiple gene edits with high precision and has been applied to model organisms like Pseudomonas and Escherichia coli for bioremediation purposes. These techniques can also be adapted for non-model organisms such as Achromobacter and Comamonas to express specific genes relevant to bioremediation process. Metabolic engineering is another way to optimize microbial pathways for contaminant degradation. Enzymes like oxidases, esterases and phenol oxidases play key roles in bioremediation as seen in organisms like Bacillus species and fungi like Phanerochaete chrysosporium. Then we have the recombinant enzyme production and immobilization which enhances enzyme stability and efficiency, making enzymatic bioremediation a green effective method for degrading persistent xenobiotics like PAS, TNTs and PCBs. We can also use genetic engineering for heavy metal remediation. Genetic engineering improves the bioremediation efficiency of heavy metal by inserting metal binding proteins and peptides into their extracellular space, screening for microorganisms with strong adsorption capabilities and chemically altering the electrophilic groups on their outer surface. Genetically engineered microbes have been created with channels, secondary carriers and primary active transporters to absorb heavy metals. This list table we can see some of the genetically engineered microbes which have been used for heavy metal remediation, and then we can see in this last column the various metals which are remediated, and here we can see the different type of genes which have been either inserted into the microorganism or which have been modified. For example, in E. coli you have these PCS metalloregulatory protein, then ARSR organomercurial lyase, then mercury transporter, then we have the mice metallothionein gene. So, these are used for bioremediation of cadmium, arsenic and mercury. So, you can go through this table and read more about the various genetically engineered microbes and the genes which have been actually inserted or modified targeting certain heavy metals. Another approach is the omics approach for microbial bioremediation. So, here we have these microbes from where we can get DNA, RNA, protein and metabolites, and the corresponding fields which deal with these molecules are the DNA metagenomics, then RNA, transcriptomics, and protein proteomics, and metabolomics. So, we go for bioinformatics study and secondary metabolites and functional gene and enzymes, and then we understand the metabolic pathways and also try to modify them and optimize them to produce the engineered microbes which will be degrading the pollutants in the environment, and therefore, we obtain a bioremediation through this omics approach. Omics studies have revolutionized metabolic engineering for bioremediation by providing comprehensive data sets that allow for the systematic optimization of microorganisms to enhance pollution or pollutant degradation. In the case of metagenomics, it basically involves the analysis of DNA directly from environmental samples or the total DNA of all the microorganisms, and helps in revealing microbial diversity, gene functions, and metabolic potential without the need for culturing. Basically, this is the approach again for understanding the null culturable part of the microbial population. It has identified novel pollutant degrading genes, overcoming limitations of culture based methods, because only very small percentage of microbes are culturable. One of the example is the study of microbial communities after the Gulf of Mexico oil spill, where metagenomic sequencing identified bacteria such as Alkani-Vorax and Pseudomonas that degrade hydrocarbons. This helped accelerate bioremediation by revealing the key genes and pathways involved in oil breakdown. So, transcriptomics basically focuses on the active genes expressed within microbial communities by studying the mRNA profile. This helps in understanding the functional responses of microorganisms to pollutants, as well as identifying key metabolic pathways involved in biodegradation. For example, if a particular pollutant, say hydrocarbon is there in the environment, that hydrocarbon will impact the expression of genes of the overall microbes in that population, and many of them will be trying to express certain genes to produce certain enzymes or proteins which will be able to degrade those pollutions. So, through transcriptomics we can understand that overexpression or underexpression, as the case may be. So, some of the example include the case of Polaromonas, where transcriptomic analysis identified genes elevated in response to cis-dichloroethene, including those for antioxidant proteins and transporters. Similarly, in the case of proteomics, we examine the protein expression profiles of microorganisms which are exposed to pollutants. It identifies key proteins and enzymes involved in pollutant breakdown, offering insights into microbial physiological responses and potential pathways for enhanced bioremediation. It is important to remember that the transcriptomic profile and the proteomic profile may be different. Not the entire transcriptome may actually be reflected in the final proteome due to various events. So, proteomic analysis of Microbacterium van Baerleni revealed the induction of dioxygenases and catalase peroxidase when exposed to polycyclic aromatic hydrocarbons. Then we have the metabolomics, which studies the changes in metabolite concentrations with microbial communities exposed to environmental contaminants. It reveals metabolic shifts, aiding in the understanding of microbial activity and the environmental impacts of pollutants. Some examples like the metabolomics linked changes in metabolite levels to microbial responses to toxic substances like hydrocarbons and pesticides has been reported. Now, let us go into a brief discussion of the use of immobilized microorganism technology in bioremediation. This method enhances bioremediation by stabilizing biological cells, preventing competition with local species. It provides a controlled environment where microorganisms can break down contaminants over extended periods, thus offering higher degradation rates compared to free living microbial systems. So, we have various cell immobilization techniques. The number one may be the chemical method, the first one being the adsorption and the second one covalent bonding. Then we have the physical immobilization techniques, where we may entrap the microbial cells or we may encapsulate them. So, let us discuss about the adsorption first. The immobilization of cells through adsorption is accomplished by moving the target cells from the bulk to the support surface or the matrix surface. So, here these cells or biocatalysts are adsorbing on the surface of these matrix and not inside of these matrix. So, adsorption basically relies on the physical attachment of microorganisms on the surface of a water insoluble carrier, mediated by weak molecular forces such as van der Waals interactions, ionic forces and hydrogen bonds. These weak forces help maintain the natural structure of the immobilized cells without causing structural changes. In the case of covalent immobilization, we have a active matrix or surface where we may have a spacer molecule and a linker molecule through which the cells are immobilized by covalent bonds. This is a cost effective method of immobilization that relies on creating a covalent link between the target cells and the support matrix in the presence of a binding agent. The functional group or groups in the cells surface are covalently bonded to the support material directly or through some linker. Then we have the physical methods of immobilization, the number one being the entrapment, as we can see here in the left figure, and the encapsulation in the right figure. Cell entrapment is an irreversible immobilization technique that traps particles or cells inside of a support matrix to provide the cells with protection from outside aggressions. Encapsulation is a form of entrapment in which the cell or enzyme is contained within a semi-permeable membrane, where immobilized cells are free to float within the core space. The membrane's semi-permeability permits the movement of unrestricted substrates and nutrients. What are the advantages of immobilized microbes over free microbes? Number one is it helps in the separation of biomass efficiently, easier to separate biomass from treated water, improving cleanup and reducing post treatment processes. Then we have improved spatial temporal control, which ensures better control over the distribution and retention of bacteria in contaminated areas, avoiding washout. Then we have the enhanced survivability and the stability. This protects bacteria from environmental stressors, extending their lifespan in harsh conditions. Then we have the advantage of reusability. Immobilized cells can be reused multiple times without significant loss of activity until and unless they are viable, making them more cost effective. Then there is reduced risk of washout. Immobilized cells remain in place within the contaminated media, allowing more efficient treatment. So, we have seen that genetically engineered microbes can be very efficient in the bioremediation process, but since these are modified genetically and they are synthetic, we need to do the risk assessment. So, in this case we can see here the field applications of genetically engineered microorganisms for bioremediation process, which involves the species Pseudomonas fluorescens HK44 and it was done in situ. The risk assessment and approval for the release of GEM for bioremediation is often complex and slow, which discourage their broader use. Regulatory bodies demand detailed risk evaluations before application approval. Of course, we should not be frustrated. This is very important for our own long term benefit. So, certain concerns over species classification, gene transfer, antibiotic resistance and pollutant metabolism to more toxin forms delays approval in this particular case of Pseudomonas fluorescens. Then field trials were conducted in control lysimeter structures that, while artificial simulated natural soil conditions and provided better insights than the lab tests. So, what was the outcome of this risk assessment of these genetically modified Pseudomonas. In general, it is often assumed that jams face survival challenges due to the energy burden of foreign genes. In this case, Pseudomonas strain did not show such burden and survived in the field. However, predicting genetically modified microorganisms or genetically engineered microorganism survival in situ remediation remains difficult due to unpredictable environmental factors. And due to these, the use of genetically engineered microbes widely approved for large scale commercial use in bioremediation is still limited. So, with this we come to the end of this lecture. Thank you for your kind attention.